Hi, I'm Tina Carbrello, Chair of the NAMS Membership Diversity and Inclusion Committee, and it's my pleasure to welcome you to the next installment in our NAMS webinar series. Today, I'm pleased to welcome Assistant Professor Christine Duval from Case Western Reserve University, who's going to talk to us today about the use of membranes in nuclear medicine. Christine, I'll hand it over to you.
Great, thank you. All right, so I'll share my screen and then we'll get going. Awesome. So thank you, Tina, for the introduction. My name is Christine Duval. Like Tina said, I'm an assistant professor at Case Western Reserve University in Cleveland, Ohio. And today I'm going to talk a bit about the field that I work in, which I think is pretty emerging in terms of membrane science and technology. So this talk will be framed a bit more in terms of opportunities for membranes in nuclear medicine and some of the motivation that I see in that area. My laser pointer on too. There we go. Okay, so before we get started, I usually like to give a few reminders about nuclear chemistry. That way it's not a talk completely filled with jargon. So if we think about the model of our atom, we have our nucleus in the center and it's surrounded by electrons. Inside that nucleus is made up of protons and neutrons. Protons will determine the element, the neutrons will determine the isotope, right? And then the electrons will determine the oxidation state. So when we have an imbalance of protons and neutrons in our nucleus, that makes it unstable. When that happens, we can have radioactive decay, and there are many different types of emissions that we can have. So one example would be the alpha particle. So that's the same mass as a helium nucleus, except it's positively charged. They can emit betas. Those have the same mass as an electron, except they're originated from the nucleus, so they're beta particles and they're negative. We can have neutrons, which are neutrally charged, or we can have these photon emissions, right, where X-rays come from transitions of electrons within different energy levels in the electron cloud, whereas a gamma ray originates from an excited nucleus de-exciting. But in both cases, they still have photons as their emission. So I've separated my talk into two parts. The first, we're going to talk about radioanalytical chemistry and how they rely on extractive purification tools. So in the beginning, right, any nuclear talk usually starts with a history of the field. The very first separation that we had conceived of in nuclear was the Purex process. And so this was a method of separating the uranium and plutonium that were produced in the first sustained nuclear reaction, which happened in the Chicago pile reactor. So after that reaction, they wanted to purify out that uranium and plutonium, and this is the process that they use. So generally, this is a solvent extraction process. They would take their selective ligand, which is this tributyl phosphate, dissolve it in kerosene, and they would mix that together with their irradiated fuel. So that was uranium, plutonium, and fission products, dissolve that in nitrates, mix those together, and then you would have a phase separation. So what we would see is that this tributyl phosphate would complex with the nitrates of uranium and plutonium and migrate into the organic phase, while the nitrates of all the other metal ions would stay behind in solution. And so this was the very first benchtop way of doing this purification. And it was later scaled up into a large scale separation that actually still happens at the Savannah River site in South Carolina. So here we have continuously operating liquid-liquid extraction. Each canyon is, I believe, the length of 3 football fields. So it's hundreds of mixer-settler operations stacked one in front of the other. And this is how they do continuous, unattended, remotely operated solvent extraction. And it's the only location in the United States that's doing this. So this worked very well at a very large scale, and it was quickly developed, in the early years of the Manhattan Project. Then, right, we think forward, in the field of radiochemistry, one of the household names is Phil Horwitz. So Phil Horwitz was a radiochemist at Argonne National Lab. That's him on the left. And so once the community started doing benchtop radiochemistry, we realized we might need a different tool other than solvent extraction. And so Phil Horwitz was exploring some methods to move beyond solvent extraction, and he ended up founding a company, called iChrome. They're still in business now. And so in the very early stages, what they decided to do was purchase polymeric resins. They actually bought amberlite from Dow, They would physisorb extractive ligands into those resins, do whichever radiochemistry separation they want, right? And then at the end of the day, they were able to sell that, to other users of the technology. The way that this looks, here we go. The way that this looks are very small resin-packed columns. So I'd say they're typically, 3 to 5 milliliters. They might be the size of your thumb. And so these columns are packed with those physisorbed resins. So over the years, there's been many different flavors, and these have really formed the core toolbox that radiochemists use when they're doing experiments either in the lab or even as some of these radiochemistry industries have begun to emerge, like medical isotope purification, which we'll talk about today. So on the bottom here, right, each of these, you'll see the trade name of the resin that's been produced by iChrome. So usually when you read the literature, that's what's reported. What I put above that are the different chemical names and structures for the different organic ligands. So something you might notice, this first one, so our Utiva resin, which is our diamyl phosphonate, that has a very similar structure to our original tributyl phosphate, which was used in the Purex process. So these phosphoryl oxygens here, right, are highly polarizable, and they form nice, strong complexes with our electron-rich actinides. So you'll see, here we go, diamyl phosphate, HDEHP, right, in the CMPO. All of these feature that same phosphoryl chemistry. Some of the more emerging ones, I would say, that are gaining a lot of popularity these days as we're looking at separating trivalent lanthanides, which are also rare earths, are some of these DGA, diglycolamide functionalities. So you'll see here, we have these 3 oxygens. They tend to form a more rigid structure. Usually you get a 2-to-1 binding motif between your ligand and your ion. And so these were all, in that same way, incorporated in a physisorbed way into those resins. So here in the bottom right, I show, this is just an SEM of some of those Utiva resins. You'll see they have very small pores. There's been some effort in the same community to look at different support materials. So whereas resins might have more interconnected and disordered pore structures, some have looked into these ordered mesoporous carbons where the pores have more structure to them, but at the same time, they still have some of those smaller pore structures. So to prepare these resins, the utility of these is really that they're relatively simple to make. So if you take your extractive resins, you'd prepare them using a dip coating method. So you would just take a solution, some solvent with your ligand dissolved in it, dip your resins in there, let them soak for a little while, filter them out on a Buchner funnel, and then you're able to pack your resins into your column. So this technology really has dominated the radiochemistry field for a long time. It solved a lot of problems associated with solvent extraction. For example, we have reduced mixed radioactive hazardous waste because we don't have gallons of kerosene to handle anymore. We also eliminated some of the issues that come with liquid-liquid extraction, where you form a third phase between your aqueous and your organic phase. Forming that usually limits mass transport and makes your separation more difficult over time. These resins are great because they're modular. You can stack them, you can mix and match chemistries. And another major thing is that regenerating them is relatively simple. You can just switch whatever your feed to your column is, and you can back extract out whichever species had adsorbed onto that column. However, many times, and this is something that I think we've learned a bit in the membrane community, resins are not always the right tool for the job. So one of the major problems that we see with any resin, right, is that we have small particle sizes, and what we have is diffusion limited transport. So if you have a packed column and your species is diffusing through the interstitial space in between your resins, right, when you go to have a binding event, that resin or that ion needs to diffuse, right, not only to the surface of the resin, but it needs to diffuse into the pore to access more of those binding sites, right? And diffusion is generally a slow process, right? Whereas convection could be much, much faster. And so with these resin-packed columns, if you want to have a fast separation, you basically limit yourself to only using the surface area of your resin, and you aren't able to utilize any of that area in the inside. Or you operate at slow flow rates, that way you can get full utilization of this packed bed, but then you have a slow separation. So some of the persistent challenges that we see in radiochemistry specifically, almost every study reports operational flow rates of less than 1 milliliter per minute. We know that we have this trade-off between particle size and surface area, particle size and pressure drop, right? Smaller particles can increase your surface area, but then it's going to be harder to flow your fluid through the packed column. And finally, if you're physisorbing a ligand into your resin, over time, it's been documented that you have some leaching of that ligand from your resin. So that either limits the reusability of that material, if that's a criteria that's interesting to you, or it can end up contaminating your product. And in some applications, that's not desirable. So when we think about the radiochemistry community, a lot of our work has been done, on the very large scale using solvent extraction. We started working at the bench top using some of these resins, but now if we're interested in moving into this, more intermediate volume for our samples, we're realizing that extractive resins are difficult to scale up. They might be fine for, tens of milliliters, but if we have to move up to a liter solution, our purification time is just going to be too long. So we can take clues from the biopharmaceutical community, right? So the membrane community has been thinking about the difference between resin-packed columns and membrane columns for a long time. One of the earlier papers that I could find is this one from 1988, where Brandt went through and talked about membrane techniques that could be used for bioseparations. And here, right, they describe the ideal chromatography column as having an infinitely short bed height. That way you can maximize throughput and minimize your pressure drop. And you would also have an infinite width with total ligand utilization, right? So when we were talking about the resins previously, we were concerned about not using that center area of the resin if we were operating at fast flow rates, right? That leads to an inefficient use of your column. And so if we can try to make all of our ligand available, that way we can maximize binding and our productivity, right? When we think about that resin-packed column and we apply these criteria, we basically shrink it down into the geometry of an adsorptive membrane. So membrane adsorbers are microporous membranes. They can be coated with functional ligands that selectively interact with your desired analyte. So sometimes that desired analyte is your product. So we're talking mostly here about catch and release or bind and elute chromatography. So here, in this version of things, you can consider flowing your solution through. You would have your membrane, right, with perhaps these polymeric ligand functional groups coming off of the surface and pores. Your desired product would interact with those ligands. It could be retained. The undesired impurities could pass through. You could also think of another, so they would pass through. And then you would take a second step where you would want to collect that product, right, and you would elute it off of your column. So I think there's a common misnomer between, I think, the chemistry communities and the membrane community when we talk about membrane chromatography. So here, right, as opposed to our silica-based chromatography columns where we're looking at a difference in mobility down the column, here it's truly a catch and release kind of collection. So you could also imagine an alternate scenario where you might be scrubbing out the undesired product and allowing the desired things to go through. In both cases, those would be adsorptive membranes. Okay, so there's a wealth of options for these technologies that have been in the literature for membrane adsorbers in the biopharmaceutical area. I'm just going to highlight two because this is a nuclear medicine talk, but I just want to give an example of how membrane adsorbers are typically made. So in this first example here, what they did is they took a microporous regenerated cellulose membrane, went through, modified that surface to implant, or basically to install surface initiators. So you can start ATRP to graft these poly-GMA brushes from the surface of the membrane. And then they went through a second step to install their functionality, which is this multimodal ligand here. So in this case, you can see we would have polymer brushes. From that polymer brush, you would have your selective ligands dangling off the polymer chains. And in this case, they were looking at antibody purification. So the analyte there was IgG, this immunoglobulin G. There's been other attempts at making these membrane adsorbers. So in this case, they went through and electrospun their own membranes. So these were polyacrylonitrile membranes. The surface was activated. UV polymerization was used to grow this poly-GMA brush. And then after that, different ligand functionality was added, in this case, to make a weak anion exchange membrane. Right? And then in this work, the target was going after a model protein, which is BSA. So outside of just the academic literature, right, membrane adsorbers have been around and implemented for quite some time. I think 10 years ago, it might have been, if you were looking at processing techniques within biopharmaceuticals, membranes had, the highest growth, the highest growing sector within that business type. So here we can see we have, companies ranging from small startup companies all the way up through larger ones like 3M and MilliporeSigma. They have a variety of products out there. And what I think is most interesting to me, right, is that we can see they all range from smaller syringe filter type columns all the way up through, these tall, many liter purification systems that you can see. And when they're used in industry, right, they're operated completely in sterile environments. And they're able to, they're used basically in a combination of upstream and downstream processing. So the potential is there, right, for these membrane adsorbers to make it out into industry. So I think this, for me, is one of the more powerful pieces of evidence that we have. So typically in the adsorber community, we always say, we have our catchphrases of, resins are diffusion limited, membrane, their transport is governed by convection, and so you can get these higher flow rates without sacrificing product recovery. So I feel like that's been our mantra for a long time. And then recently a paper was published that went and did a very clear head-to-head comparison, at least in the biopharmaceutical industry, looking at a membrane and a resin-packed column with similar chemistries, looking at the same analytes and going through and actually doing this head-to-head comparison. So what we can see here, we're going to be comparing the sardobind Q membrane. So the ligand type is quaternary ammonium. The analyte that they were interested in is this BSA protein. And the membrane had a 3 milliliter bed volume. So if you take the analogous resin-packed column, right, which is this Q stepharose resin, nearly all of the criteria are identical. Where we see a big difference is the bed height, but we know that because that's a benefit of membranes versus resins is the geometry. And so you can see with the membranes, right, is that we're going to expect convective flow to happen. So for that ion to enter the membrane column, it will enter the column, it will diffuse to the binding sites, which are those ligands decorated on the surface, right? And we expect that process to be fast, as opposed to our resins, where we can expect that we will have flow in our interstitial spaces, but as we start binding to our resin, right, we're going to have that diffusion limited process of the ion entering the resin. So to be able to evaluate these and compare them head to head, usually what we do are a series of experiments called breakthrough curves. So in these curves, what you will do is monitor the effluent concentration coming out the bottom of your column, right? And then we normalize that to our inlet concentration that is entering our column. So what that means is if we have a value of 0, everything is sticking to our column. If we have a value of 1, then we have complete 100% breakthrough through that column. So what we see here for the resin case, they started flowing through their BSA solution, and they did this at several flow rates. So you would see if we follow the black line, which is the lowest flow rate, we start introducing the BSA, right? And we have nearly perfect capture. All of that BSA is being retained until a certain point where that BSA begins to break through, right? And so it's coming through in the effluent, and then eventually we reach 100% breakthrough, and that represents a point where the column has come to an apparent equilibrium with that feed concentration. So that is what it looks like for our resins. And what you will see, right, is as we increase our flow rate, that time to break through gets shorter and shorter. So we go to faster flow rates, and what we see is that the BSA is not able to diffuse into the resin, right, and into those binding sites to be able to be retained. So it's just exiting the column, right? So that is considered lower product capture as you increase your flow rate. When you repeat that experiment, but you use the membrane adsorbers, what you see is that if we follow the one single line, right, we follow our line, we're retaining our BSA on the column, it eventually breaks through and we reach that 100% breakthrough. But what's most interesting about this is as we increase the flow rate through the membrane adsorber, our breakthrough curve doesn't change, right? So we can go up to pretty high flow rates, right? So here we go all the way up to 15 milliliters per minute and we see that there's no difference in that breakthrough curve. And that's because the mode of transport that dominates in these membrane adsorbers, right, is convection, whereas our resins are limited by diffusion. So this is the backstory to our tagline, which is product capture in membrane adsorbers is not a function of flow rate. And that means practically that these membrane adsorbers can operate at higher flow rates without sacrificing product capture. So that makes them really attractive for doing faster separations. Okay, so I'm going to leave that thought in your mind. And the next thing that I want to enter into, right, is kind of this need for new purification methods in this area of nuclear medicine. So to tell a complete story, we're going to focus on specific medical isotopes or radiopharmaceuticals that are used to treat prostate cancer. So in nuclear medicine, right, that decay pathway dictates how the isotope is used. So we can have therapeutic medical isotopes. So these have short range emissions. So either alpha particles, beta particles, Auger electrons. Some of the more famous ones are listed down here in case you're interested. And we call those therapeutic. We can also have diagnostic medical isotopes. So these have longer range emissions. So those could be species that emit gammas inherently. They could be species that emit positrons. And then just like in PALs, right, if those positrons annihilate, then you can create two equal energy gammas that can be used for detection. So that's what we have here, right? So for these, if you've ever had a PET or a SPECT scan, you were injected with what they call a contrast agent. That contrast agent is radioactive. So therefore you are radioactive while you're getting imaged. And you'll be, slid into this detector here. And this is a bunch of gamma cameras that are surrounding the patient. So you can start doing that sort of diagnostic imaging. There's also a really fun newer class of medical isotopes that we call theranostic because they're both therapeutic and diagnostic. And these are species that can have short and long range emissions. So maybe it emits a beta, but it has a coincident gamma that comes off of there. In that case, you can use it for both functions, which makes it really easy for the radiochemists because they only need to use one sort of coordination chemistry to be able to send that into a human. Okay, so first we'll focus on radiation therapies that can be used for treatment. So in this case, we'll focus on prostate cancer because that's where a lot of the initial research has been focused in this area. So ionizing radiation can damage cells, right? So we know this from historic incidents where we've had either, nuclear accidents or, in World War II when we dropped the atomic bombs. We have a lot of data where high doses of radiation can lead to cell death. Ever since, I think, the 1950s or so, there was a famous Atoms for Peace speech that was given at the UN, and that kind of announced that, we know we can use nuclear for bad, but really we should be trying to use nuclear for good. And since then, there's been a concerted effort to look into nuclear for power production, and then nuclear in medicine has really sprung out of that. So at much lower doses, ionizing radiation can be used to selectively destroy cancerous cells, which are ones that we would like to clear out. So the way that happens is the ionizing radiation enters the cell, right? And our cells are primarily made of water. So what ends up happening is most of the time we have something called indirect action. So that alpha, beta, or gamma will ionize the water and it will create these reactive radical species. Those reactive radicals will go and attack the DNA in the cell. And so if you have a rate of destruction that is faster than the rate at which the cell can repair itself, the cell will die. So this is the principle that is used in every type of radiation therapy. Typically, people start, so I think the more established radiation therapies are these external beams, right? So here in the center, if you have external beam therapy, a prostate cancer patient would have to go in 5 days a week for several weeks. And then what would happen is they would lay down and they would be irradiated with an external source of radiation. So that could be a proton beam or X-rays. And what would happen is the radiation would, irradiate the patient. Some of that, so if you see here, we have the prostate highlighted. So some of those cells would be cancerous, right? And they would be irradiated and those cells would die. But unfortunately, you end up irradiating a lot of healthy tissue in front of and in back of the affected area. So this is generally effective, but every time that you irradiate healthy tissue, that's what leads to things like radiation sickness and a lot of these poor patient outcomes in terms of side effects. So another option that's commonly used, right, is brachytherapy. So in this case, the patient would have these radioactive pins. So in this case, this is iridium-192. That's a beta particle emitter. Beta particles go about 4 millimeters in tissue. So they would be directly implanted into the affected area. And then that patient would be able to walk around and then, constantly receive their radiation treatment throughout the day. So in cancer treatment, and this is true for the targeted therapies that we're about to talk about, the human body is incredibly complex, and every single type of treatment, will have a different patient outcome depending on the person. So when we talk about radiation therapies and even more broadly cancer treatment, it's really important to have many tools in the toolbox. So in this case, perhaps brachytherapy is a little more targeted than the external beam therapy, but at the end of the day, it's all about choosing the treatment that's going to work best for the patient. So another approach that has been taken, and this is really the area where we're going to start talking about separations. An area where there's been a lot of growth recently has been this idea of targeted therapies, right? So these would be injectable radiation therapies that will selectively transport to the affected area. So the very first one that was ever FDA approved, and this is the one that I think is kind of like the flagship drug that a lot of the newer clinical trials are referencing to say, well, Xofigo made it through, and so there's possibilities for these other drugs. So this one's manufactured by Bayer, and it is a radium dichloride. So radium-223 is an alpha emitter. The alpha particles only go about 40 microns in water or tissue. And the way that it is administered is a 1-minute injection. You would come back for multiple treatments, right? And then it would be administered over the course of 5 months. The way that the drug is targeted to the cancerous region is very specific. And so this drug ends up being limited in its use. So here, radium and calcium are very chemically similar, right? They're both divalent ions, and they tend to bioaccumulate in the same area, which is the bone. So if radioactive radium is injected to a patient who has a bony metastasized cancer, for example, a prostate cancer that is spread to the bone, those cancer cells are going to be growing at a faster rate than the healthy bone. And so the radium, right, is going to be taken up very quickly into that area. Once it's incorporated in the bone, right, it's localized and it can locally irradiate those cancerous cells. So this has shown pretty good patient, so it's shown pretty good outcomes for patients in terms of pain management. It generally extends the life of the patient by about 30%. And so to put this in perspective, because radiopharmaceuticals, I think the field operates on a very different scale. We think about how much radium-223 is needed for a specific dose to be administered, right? We can look at doses are prescribed in terms of radioactivity per mass of the patient. But at the end of the day, right, each injection of this Xofigo drug is only about 5 nanograms of material, right? So we're going to start talking about purification and scaling up, but just want to put it in perspective, we're not talking about, millions of gallons per day like we would in commodity water treatment. But here we're going to be talking about recovering the needle in the haystack from relatively small batches of these specialty radiopharmaceuticals. Okay, so you might ask yourself, what if we want to treat a cancer that isn't in the bone, right? So radium targeting bone is one example. Iodine naturally accumulates in the thyroid, so radioiodines are often used to treat thyroid cancer. But what if we wanted to choose where in the body this was going to go, and not just based on the natural metabolism of the body, right, but more based on targeting metastasized cancers? So the way to do that is with these targeted alpha therapies. What you would do is you would take the radionuclide and you would conjugate it to some biomarker or biovector that can help you seek and destroy within the body. So this is part of a wider blanket of personalized therapies. So you could imagine, right, that you can use a similar targeting agent and attach it to a small molecule or protein that's toxic to the cell, right? That's how some chemotherapies are done. You could attach these cytokines that just sound the alarm for the immune response to come to that specific area. But as radiochemists, we're interested in this radionuclide therapy. So some of the most famous ones, the very popular ones right now. So we've mentioned, right, radium-223, which is not conjugated, but just naturally goes to the bone. Other ones that are interesting are these alpha-emitting actinium-225 and thorium-227. So they have similar half-lives to that radium-223, right? And so the half-life is related to your rate of decay. So we always want to be keeping in mind that it's a game of competing rates, right? We want enough alpha particles to be produced per time such that we can destroy that DNA before it repairs itself, right? But we don't want to have so much of a radiotoxic dose that we do more damage than we actually want to. So the sweet spot for that, for these alpha therapies, seems to be in the window of 10 to 20 days. So to actually send it into the body, there's a couple parts to the drug. So the first one would be taking your radionuclide, and we need to conjugate that and chelate it within some chelating group, right? So famous ones are DOTA, NOTA, and DTPA. So this is DOTA shown here. And so these macrocycles are slow complexes to form with the ion, but then they are kinetically stable. So they will hang in there for a long time, and they remain stable when they go into the body. And then here, the other part of the drug is the biovector. So in this case, I showed an antigen. This is one of the PSMA antigens for prostate cancer. So that's prostate specific membrane antigen. And so this will go and locate any metastasized tumors that would have started from the prostate. So they'll all have the same chemical signal on their outer membrane. And so this can help, seek out those areas to locally irradiate those cancerous cells. So these drugs have shown great promise in clinical trials. So actinium-225 PSMA, this is one of the most heavily studied ones, and I think the one that is closest to commercial viability. So what you can see on the left is a scan of a patient before treatment. You can see that cancer, those dark spots have metastasized and really spread throughout the body. After treatment, you can see an incredible reduction in those tumors, right? And that is because of this localized action of the targeted alpha therapy. So after 24 weeks, the majority of the patients who participated in this trial saw a drastic reduction in tumor size. And the primary side effect here, right, instead of very intensive radiation sickness or some of the other side effects that we see with like chemotherapy treatments, the primary side effect here was a very aggressive case of dry mouth. So it's not your normal dry mouth, but it is, I would say, one of the milder side effects that you can have from a cancer treatment. So, seems great. Unfortunately, what we have is a supply issue when we think about our medical isotopes. So most medical isotopes are derived from legacy waste in the US. This is just how the research cycle goes. From time to time, researchers will go in, harvest out different isotopes, purify them. Perhaps they go to a clinical trial and we realize that they're useful. And then we say, oh no, this isn't a sustainable supply. So actinium-225 fell into that exact trap. All of our actinium-225 is derived from stockpiles of weapons-grade uranium. So the US used to make, so in addition to uranium-235, which we use for weapons, you can also use uranium-233. It's also fissile. So at Oak Ridge, over the years, they had accumulated a lot of legacy waste from uranium-233 production. And so there's a stockpile of thorium-229, which is part of that waste. So in this stockpile, thorium-229 is slowly decaying to radium-225, right? And every so often, Oak Ridge will go in, harvest some of the radium-225, right? And then they will use that to generate our actinium-225. Unfortunately, right, as you can imagine, there's first of all a limited supply of that uranium-233. We're not making it anymore, and there's a lot of national security efforts to downblend stockpiles of uranium-233. And then on top of that, at our current production rate, right, there's only enough to supply 1 in every 5,000 treatments. So for every 5,000 people that qualify, only 1 of them can actually get this. So yeah, so every so often, Oak Ridge will go and harvest this actinium-225 through this scheme, right? And one of the reasons that this actinium-225 is so potent is because we have a 4-alpha cascade that comes after it. So the half-life of actinium-225 is 10 days, and then all of those decay products are very short. So if you can bring that actinium-225 close to the cancerous cell, you essentially get 4 punches in a row where those alpha particles can go ionize the water within the cell and then have it go and destroy some of the DNA. So the next logical question would be, how can we get actinium-225 if not from weapons grade uranium? This is where the chemical engineering starts to come in. So just like any other chemical reaction, or if we want to keep drawing the analogies to the biopharmaceutical industry, right? We can make our monoclonal antibodies or whatever it may be. We can do it in our bioreactor. You can grow them in mouse models, right? So in some way you have to produce the desired product. And here in nuclear medicine, we do that in either a linear accelerator, which is used for heavy nuclei, or some of the lighter nuclei can actually be produced in biomedical cyclotrons in the basements of hospitals. Actinium-225 is very heavy. It's an actinide. And so the energies you need to bombard, to bombard your targets ends up being pretty high. So these are usually made at national labs. So what you would do is you would take your target material. This is just a metal foil. It can be, the 2 primary routes for actinium production are taking radium-226 or thorium-232, and then you bombard it with protons. And so what these nuclear reactions are describing are your reactants here, so the radium and the proton, and then your products are on the other side. So you would get 2 neutrons out and then some actinium-225. So what comes out of your linear accelerator then is your irradiated target, which has your unreacted target material, a very small mass of your desired product, and a lot of undesired byproducts that you don't want, that are just a result of the fission reactions. So it becomes a purification game. So at the end of the day, we want to isolate our desired radioisotope. That way it can be attached to one of those drugs and it can be injected into our patient. The way that that purification happens right now is through a series of resin-packed columns, right? That is the industry standard for most radiochemists. They're, like I had mentioned before, infinitely better than working with solvent extraction in the small scale, but we will see that we have a really large bottleneck associated with that purification process. So the way that it works is you would take a strong acid. And when I say strong, I mean something like 10 molar nitric or hydrochloric acid, sometimes a touch of HF for good measure, dissolve that target, and then it goes through the series of columns. So we're doing highly acidic separations. We're also working in a radiation field, which can present some materials challenges. And like I mentioned, ideally we separate out our desired isotope. We would also separate out unreacted target materials. Ideally, these are recycled because those are isotopically enriched material, is labor intensive and expensive to make. So we would want to find a way to bring that back in our process. And then we would have a bunch of undesired fission products that we can either dispose of or repurpose for other things. So for actinium-225, the major species we need to worry about purifying are these three right here. So whether we start with a radium or a thorium target, we're going to co-produce the other, and we'll want to fish out our actinium product. What ends up being really interesting about a lot of these separations that result from nuclear reactions is that if you're bombarding with protons or even bombarding with small nuclei, right, usually the reactions that are accessible just by the probability are a lot of the neighbors on the periodic table. So usually your separations are, 3 in a row. And if you're doing that for lanthanides, it becomes tricky because lanthanides are very chemically similar. If we're doing it here across some of the actinides and then radium, we have a little bit of benefit of they have different oxidation states. But all of those things really get factored into designing these specialty separation materials. Okay, so the state-of-the-art purification scheme, this is what's used for, to my knowledge, right, every actinium-225 PSMA drug that has been injected into a patient for clinical trials has experienced this long purification process. So usually what happens is you would dissolve 500 milligrams, right, we're talking, this is our generally small scale, 500 milligrams of our irradiated target and 75 milliliters of our strong acid. It's then diluted up to pH 2, and they add citrate. And this process here is relatively simple because it only uses 2 primary columns. So in the first column, we would load our sample onto the column. And what happens in this step is at low pH, like pH 2, thorium forms an anionic complex with citrate. So we have this large, bulky, complex ion that contains thorium, right? We have our cation exchange resin here. And so that large thorium is not going to want to stick to the column, and that's going to pass through. So this is our thorium debulking step. And our target was thorium, so that's going to be most of it. So this takes quite some time. Next, what you do, you start switching your feed conditions to try and elute certain products off of your column. So first we'll switch to 1 molar nitric acid. That should wash out some of our divalents, so our radium and ruthenium. Then we'll come through and elute with 6 molar nitric acid, and that will kick out our actinium, which is desired, our cerium, which is chemically similar to actinium, right, and any radium and barium that we have left over. Then we move on to our next column, right, and this column here uses that famous DGA chemistry, right, the diglycol amide is absorbed in the resin. So we load our entire sample on the column. Some of the radium and barium wash through. We wash the rest of the radium and barium out. And then what we're left with is our DGA resin complexed with actinium and any of the lanthanide products that are remaining. So in this case, it's primarily cerium. So then we switch again with another strong acid, and then that's how we actually pop off our actinium. So this process, as you can see, requires many different feeds. It has 2 different columns. We had discussed that the flow rate through these columns, right, is usually less than 1 milliminute. In this paper, they actually reported their flow rates, and it was 0.5 mil per minute. So what this means is that to isolate 26 micrograms of actinium, right, it takes about 6 to 8 hours, and that's just for this purification process. That doesn't include quality control. That doesn't include any of the waiting time that you have after you stop bombardment before you're actually able to purify your sample. And so we really have a bottleneck in our purification process. So this is one of the areas that there's actually a US Commission on Isotope Research and Development, and they've identified not only new production methods, but new chemical processing methods as a major bottleneck in our ability to bring these types of isotopes into clinical trials, and then also once they pass clinical trials, being able to have a sustained supply throughout the United States. So to scale this up, right, this is 6 to 8 hours for 26 micrograms. If you want clinically relevant quantities, we need to scale up 10 to 100 times. And if you think back, these alpha-emitting radionuclides, they're so useful and so good at treating cancer because they have short half-lives. So if you have something that has a half-life of 10 days and you're taking, a day to purify it, right? You're losing a significant amount of your product just because it's decayed away, right? And we already have to start with, calculating our quantities back from, so there's producing it, right? And then there's also shipping time that it waits at the hospital, time for it to be injected into the patient. So if we want to maximize the amount that can eventually get to the hospital, right, we really need to be maximizing how much is coming out of our process here. So minimizing those purification times. So this is where I think the opportunity for membranes really is. And a lot of it ties back to some of those lessons that we've learned from the membrane adsorber community, right? Transitioning from these resin-based purification methods that used to exist in biopharmaceuticals and moving forward into membrane-based ones. So radiochemistry, in my opinion, right, is a new frontier for membranes. This area is very new, I would say, in the traditional membrane sense. So this is an example here. This is a paper from my group, but we started looking at radiocopper, which is one of those theranostic isotopes, right? So going through some of these same mass transport arguments that we've seen in the biopharmaceutical community, but how would we start considering ion selectivity in these materials? And how do the design considerations change when you're not filtering a large protein, but you're filtering ions from each other? Membranes have also started showing up in other aspects of radiochemistry. So this work actually came first. So there's this interest in using membranes as a sample preparation platform for different nuclear forensic analyses, right? So if you're looking at uranium or plutonium in places where they shouldn't be, if you can collect and concentrate those on membrane surfaces, you can then use them directly in a lot of radiation detection equipment. And then that information helps inform stakeholders as to, what was the intended use of this material? Was this supposed to be here or not? So that's another area where they've popped up. And then I included another example here. I think the first logical leap in membrane technology, and maybe there's some debate on how well liquid supported membranes fit into traditional membrane separations, but I think the first step was to take from extractive resins and then take those same ligands, physisorb them into either meso or microporous inert membranes. And then in this type of work, what you would have is your feed solution coming in, carrying in your ion. It would enter this extractive ligand solvent mixture here, right, diffuse across, and then it would be stripped on the other side. So these membranes have been used in a variety of applications outside of nuclear medicine over the years. The radiochemistry community has generally become interested in these because they're looking for faster ways to purify their samples. So I put an example in here of a group that was looking at isolating yttrium-90, which is a beta-emitting medical isotope, from strontium-90, which is something that you don't want to use as a medical isotope. So there are seeds of membranes in the community, but I think the need is great, and this would be an excellent area for membrane scientists to start to enter. So there's some opportunities and challenges, right? So much like the biopharmaceutical industry, the radiopharmaceutical industry can really benefit from innovations in membrane separation technology. In particular, membrane adsorbers are promising because that product recovery is not a function of flow rate. And there, maybe there's some debate in the biopharmaceutical industry whether or not they need to go to extremely high flow rates. I'm not sure that it's as necessary for them as it would be in radiopharmaceuticals where your product is decaying away. So you have a legitimate time constraint that you need to consider when doing your purification. And so budding challenges, of course, are going to be, how do you choose your ligand? What is the appropriate ligand ion selectivity? The kinetics of those interactions are going to dictate the flow rates you can actually work at. So despite any mass transport arguments you might be able to make, if the complexation is too slow, you might be limited in your flow rates. And then other unique design considerations that we have in our field are really related to long-term radiation stability of these materials, and other design considerations that come from working in pretty strong acids. So one of the earliest things that we started thinking about is, beyond the typical supports that are used in biopharma, which might be cellulose or regenerated cellulose or, polyether sulfones. What kind of base materials could you possibly use that would be stable in, 10 molar hydrochloric or nitric acid? So, yeah, I would like to thank everyone for their time. And thank you to NAMS for inviting me to contribute this webinar to the series.